process of fertilization, the development of the zygote, which is the embryo, then from the zygote to the embryo to the fetus. And then the next section is the implantation, gestation, and the role of the placenta. You need to know the definition and of implantation, the role of estrogen and progesterone in maintaining pregnancy, and the structure of the developing fetus in the uterus using a diagram. And then the functions of the following parts: chorion and chorionic villi, amnion, amniotic cavity and amniotic fluid, umbilical cord, including the umbilical artery and the umbilical vein, and the placenta. Okay, so this is what you need to know. When you are studying this section, you need to keep this next to you, and then you need to tick all the boxes when you've studied each thing so that you can make sure that you study everything that needs to be that you need to study. Right, so let's go into the terminology that we need to understand for this section. As they said, you need to know the definitions of copulation, fertilization, and implantation. So I have them all on one slide here for you. Copulation is the introduction of the male sex organ into the female sex canal, followed by a discharge of semen. And fertilization is the fusion or the joining or the coming together of the haploid nucleus of the sperm cell with the haploid nucleus of the ovum to form a diploid zygote. And implantation is the attachment of the fertilized ovum or the blastocyst to the wall of the uterus at the start of pregnancy. Okay, so these are the definitions that you need to know. Make sure that you know them and you understand them, and then you will see that we're going to be using these terms in the lesson. Okay, so before we get here to this part, let's just talk a little bit about what we discussed last week about the menstrual cycle. So if you can remember correctly, we said that the menstrual cycle is a 28-day cycle, and one major event that occurs in the menstrual cycle is that on day 14 ovulation occurs. And ovulation is when the ovum is released from the ovary, right? And then we spoke about how the fimbria of the fallopian tube is going to pick up the ovum, and that ovum is going to travel along the fallopian tube via the cilia. And then it's going to stay in the fallopian tube and it's going to wait for fertilization. Okay, remember we said that last week. And then we said if fertilization does not take place, then menstruation is going to occur, and we're going to have the shedding of the endometrium, and then the ovum is going to be expelled with the endometrium. But now, when we're talking about fertilization, we are talking about if fertilization happens at that point when the ovum is inside the fallopian tube and the sperm cells are going to swim up and they are going to penetrate the ovum. Okay, so let's look at that and let's look at what happens there. So what basically happens, they're showing us over here, there's a lot of things happening here, but let's focus on what I explained to you over here, that I said that ovulation is going to occur. The ovum is going to be picked up by the fimbria, which is that funnel-like structure, and then it's going to go into the fallopian tube, and then it's going to, at this point over here, can you see the sperm cells are there? So where did these sperm cells come from? The sperm cells are in the semen. They enter the cervix, they pass through the uterus, and they enter the fallopian tube. Okay, so they're going to be traveling up here. Okay, and then the sperm cell propels itself by means of a tail using energy from the mitochondria and fructose from the semen. Now remember, we spoke about the accessory glands of the male reproductive system. The accessory glands are going to secrete a fluid, and that fluid is going to form part of the semen, and something that's going to be contained in there is fructose. Fructose is the nourishment that's needed for the sperm, right? And then the mitochondria, remember it's in the neck region. The mitochondria provides the energy for the sperm cell to propel itself, basically to swim, to move, okay? Remember it's for motility. So it's going to move because the tail is going to propel. Going to move like in this direction, and it's going to move, help the sperm cell to move up towards the ovum. Then what happens? Now it's showing us something is happening here. Can you see that? Okay, you can see over here that something is happening, but we're going to go into more detail in the next slide. So what happens now is that the sperm continues to swim with a spiral-like movement towards the ovum in the fallopian tube. The head of one sperm cell penetrates the membranous barrier around the ovum, which is made possible by the enzymes from the acrosome. And just for more information, those enzymes are called lytic enzymes. Okay? So, then the sperm is going to lose its tail and only the head enters the ovum. So let's just have a look at what's happening here. You can see this is in this diagram, this is the first diagram, and then this block over here is showing us that the next diagram is going to be an enlarged version of that block. So let's focus on this. Okay. So what we can see here, let's remind ourselves what the acrosome is. Okay, the acrosome in this instance is in this head region, it's the black region. And that acrosome is going to have enzymes, which are called lytic enzymes. And they are basically going to dissolve the barrier. Okay, the protective barrier that is, surrounding the ovum. Okay, and then it is going to enter. Okay, only the head enters. We can see over here that only the head is going to enter. The mitochondria and the tail do not enter. So it's only the head that has the nucleus. That is the only part that is going to enter the ovum. Okay, the mitochondria, or the neck region with the mitochondria and the tail, that is not going to enter. Okay, so later on we're going to learn more about this when we're doing evolution towards the end of the year, where they actually speak about how you need to know that the mitochondria is not, the mitochondria of the sperm is not forming part of of fertilization. Okay, so the next thing that happens then is that membrane. So now the membrane was broken. Okay, we spoke that it was dissolved by the enzymes, and that allowed the head to enter, and then the other part falls away. But now we need to prevent other sperm cells from entering. Okay, because remember, the important part that we need to remember about this whole fertilization is that we have a haploid ovum or haploid nucleus in the ovum that needs to fuse with the haploid nucleus in the sperm cell. So if they are both haploid nuclei, that means they have half the number of chromosomes. Then they're going to fuse, and then they're going to have the full set of chromosomes. Right? So we don't want other ovum to be coming in there now and messing up with our chromosome number. Okay, so now what's going to happen is that membrane becomes impenetrable to other sperm cells. That means that other sperm cells will not be able to break the barrier. And then the nuclei of the male and the female fuse, and a diploid zygote is formed. Okay, so now that entered, the two nuclei are going to fuse, the barrier is formed so that other sperm cells cannot enter, and the nuclei fuse, and then we're going to have the development of a diploid zygote, which is shown in this picture over here. That's showing us, no, this picture is showing us the sperm and the ovum nuclei are, so here's a nuclei here, here's a nuclei here, and they're showing us that they are fusing. Okay, it's actually a very nice little micrograph. Right, now we've got the development of the zygote. So what happens now? This zygote is now a diploid zygote, right? So how does the zygote actually develop into an embryo, and from an embryo develop into a fetus? Okay, because obviously this cell now, this is now a structure that has how many chromosomes? It's going to have 46 chromosomes because it's a diploid zygote, but now it's got to go through processes so that it can develop further. And it goes through a process called mitosis. Okay, so you'll remember mitosis from grade 10. Mitosis is needed for growth, okay, for repair, but in this instance, it's specifically meant for growth. Because what's going to happen is that it needs to divide. The cells need to divide, and they need to divide, and they need to divide, so that the structure can actually grow and become bigger and more developed. So the zygote is going to undergo mitosis to form 4 cells, and then 8 cells, until it forms a solid or a ball of cells called the morula. And then through further mitosis, it's going to form a hollow ball of cells, which is called the blastocyst or the blastula. And then that blastocyst or blastula is going to move along the fallopian tube until it reaches and implants in the endometrium of the uterus. Okay, so let's just look at the structures over here, what I said. So here we have a zygote. Let's look at the bottom picture first. We've got the zygote, and then it's going to go through mitosis, 2 cells, 4 cells, 8 cells, and then it comes into a solid mass or a solid ball of cells called the morula, and then a hollow ball of cells called the blastocyst. So I just want to put a little arrow here just to indicate to you that everything that is going on here is going to be mitosis. It's important that you know that it's mitosis and not meiosis over here. Now let's look at what's happening in the picture. They're showing us, this is just showing us the ovulation again. It's then showing us the fertilization that's happening here. And then we're going to have the 2-cell stage, the 4-cell stage, the 8-cell stage. Can you see how it's carrying on until we've got this, the modula, which is the solid mass or the solid ball of cells, and then we've got implantation over here where we can see that the blastocyst is now implanting into the endometrium. Okay, very important that you know this. They love to ask this question. They like to ask you that they can show you a picture of an ovum, they show you a picture of one of these. They're not going to give it to you in the same order. They might not give it to you in a particular order. They could give it to you mixed up, and they could ask you in what sequence they're going to develop in, and then you need to put them in order. So there's different ways they can ask this. It's very important that you know the different stages, when they occur, and how they look. Now we come to implantation. Okay, the blastocyst embeds in the endometrium, and the process is called implantation. I just want to also mention to you grade 12s that it is a blastocyst and not a blastocyte. Okay, so some learners find it easier to say blastula. That is also fine. We accept blastula, but not blastocyte, because it means something else. You will be marked wrong if you say blastocyte, because it's not a blastocyte, it's a blastocyst. Okay. So this is just showing us a little picture of what's happening over here. Can you see that something has happened now to this blastocyst? The blastocyst picture that I showed you, it was round and it was like, you know, smooth. But now this one is showing some structures developing over here, and it's showing us how it is now penetrating the endometrium and embedding into it. Okay, I'm going to explain to you exactly why and how this is happening. So you can see over here that there's something happening, excuse me, something is developing inside there. We have the embryo that is developing inside, and we have these structures, okay, on the outside. I'm going to show you now how the structures are actually, what's actually being formed here. Right, so in the beginning, even before this, now as you can see in this picture, there's a little baby there in the center, okay? But before we get to that, when the blastocyst is implanting, what's going to happen is that it's going to embed into the endometrium, right? And then it's going to form extra embryonic membranes. And those extra embryonic membranes are called the amnion and the chorion, okay? The amnion, the chorion is the outermost layer. It's covering, and it's got finger-like projections which are going to extend into the endometrium, and they are called chorionic villi. Okay, then the next layer is called the amnion. Now I quickly want to go back to the previous picture to show you. So they are showing us, like over here, they are not showing us exactly the chorion here. It is over here. It is the orange section, okay, and that is the chorion. Okay, so it's the outermost layer of this blastocyst now, the chorion, and that chorion is going to develop finger-like projections which are called chorionic villi. Okay, so those chorionic villi are forming part of the fetus or the embryo, right? And then the next layer is called, the next extra embryonic membrane is called the amnion. The amnion has an amniotic sac which is filled with amniotic fluid. Okay, and then these, now let's go back to the chorion. These chorionic villi are going to extend, they're going to become larger, they're going to become bigger, they're going to become more vascular. Okay, which means that they're going to be filled with blood vessels. So as they're going to become larger and bigger, they're going to penetrate deeper into the endometrium so that the embryo is actually fixed there. Okay, so that's the part that I explained. Now you can see in this picture, you can see the chorionic villi. You can see it's labeled over here, chorionic villi. And can you see that it's going into the endometrium? You see here's the endometrium. This darker pink color is the endometrium. And the chorion are basically projecting into there, penetrating into there. So the endometrium together with the chorionic villi is what makes up our placenta. This region over here is the placenta, it's the endometrium, with the chorionic villi that's getting bigger, more vascular and larger. It's going to be called the endometrium. I mean the placenta. Okay. Then we also need to know, you also need to know some other structures which we are going to go into now. But for this diagram, I just need us to understand, we can come back to it later, but for now I just want you to understand how the implantation is occurring. Okay, then this is just another diagram to show you the chorionic villi, and it shows you the umbilical cord. The umbilical cord, remember, is also coming out. It's the part, the umbilical cord is the part that joins the fetus to the mother. Okay, we're going to go into what it does just now, but this is how the implantation occurs. Okay, so now let's talk about the placenta. So now as this embryo is going to be developing, the placenta is now going to be taking over it, so it's going to be carrying out its function. Okay, so how does it do this? So there are large spaces which are called sinuses that develop into the endometrium. Okay, and what we have over here is that, as we showed you in the previous picture, sorry, I'm going back and forth, but the best way to show you that this is the uterine wall, right? So this is coming from the mother. And then we've got blood vessels that are going to extend here. These are the mother's blood vessels, okay? And the fetus's blood vessels are going to be here in the chorionic villi. So what happens is that these blood vessels, they bath the chorionic villi, okay? So they're just surrounding it. The nutrients are going to diffuse from the blood vessels of the mother to the blood vessels of the fetus, okay? That's how it's going to work. But the blood itself never mixes. Why do we say that? Because remember, blood flows in blood vessels. The blood does not come out of the blood vessels. Blood is flowing in blood vessels, okay? And then the nutrients diffuse from the maternal blood across the membranes into the fetal blood. Okay, so that is what is basically happening over here. We have the maternal blood and we've got the fetal blood vessels. This picture is showing us very nicely the maternal part of the placenta, the fetal blood vessels, the maternal blood, and then we also have the maternal blood that is in these spaces, right? In these sinuses. So they are in these spaces in the endometrium. So they do not mix with the blood of the fetus. That's very important for us to understand. Okay, and this is just showing us a real life picture of the placenta that is attached to the umbilical cord. Okay, so let's go to the functions of the placenta. It's the point of attachment of the fetus to the mother. It allows for diffusion of nutrients from the mother to the fetus. Okay, that's what we spoke about. Diffusion of the nutrients from the mother to the fetus. Okay, the mother is the one that has the nourishment and provides the fetus with the nourishment. It allows for the diffusion of oxygen from the mother to the fetus and the diffusion of carbon dioxide from the fetus to the mother. Okay, so the fetus is going to go through going to go through processes, metabolic processes, and it's obviously going to start giving off carbon dioxide, and that needs to go to the mother so it can actually be expelled out of the mother's body. It allows for the diffusion of waste products from the fetus to the mother, and after 12 weeks the placenta secretes progesterone to maintain the pregnancy. So remember, when we were talking about the menstrual cycle, we spoke about how the corpus luteum produces progesterone, and that progesterone is then going to maintain the endometrium. But now after 12 weeks of pregnancy, this function is going to be taken over by the placenta, where the placenta is going to produce the progesterone and to maintain the pregnancy. Okay, so these are the main functions of the placenta, but we also have protective functions of the placenta. So they also have a protective function such that they act as microfilters. They protect against pathogens, they produce antibodies, so they assist with immunity, and they maintain the endometrium. Okay, and how does it maintain the endometrium? By secreting the progesterone, okay, and also estrogen. Then we've got the amnion and the amniotic fluid. Now let's go back up again. What did we say? We said the extraembryonic membranes. There are two extraembryonic membranes, very important. They like to ask this question in biological terms about the extraembryonic membranes. So the outer one, the first one is called the chorion, which develops the chorionic villi. Then the inner one is the amnion, which has the amniotic sac, and it's filled with amniotic fluid. Okay? It is impenetrable, and it secretes a fluid in which the fetus lives and moves. So, okay, so you know when they say, when you hear that, you know, a woman would say that their water broke, okay, then what's actually happening there is that it's the amniotic fluid that is actually being expelled. So the functions of the amniotic fluid include, it's a shock absorber, so it protects the fetus from mechanical injury, thermoregulatory, prevents the extreme temperature changes, moisturizing, prevents desiccation of the fetus, and it also provides the medium in which the fetus can move and basically allows movement of the fetus. Okay, you need to know the functions of the amniotic fluid. They also love to ask this question, and really it's easy marks because you just give the functions. Okay, state the functions. Now we come to important structure called the umbilical cord. Okay, so the umbilical cord, it anchors the fetus to the placenta. So the umbilical cord extends from the placenta, which is now more the mother part, and then to the fetus. Okay, now what is this umbilical cord made up of? It's made up of one umbilical vein and two umbilical arteries. The umbilical vein transports nutrients and oxygen to the baby, and the umbilical arteries transports waste products and carbon dioxide to the mother. Okay, so you can see this lovely picture over here is showing us that there is one vein. I color coded it so that you can see that it's the vein in the picture is red, and in the notes that I have, it's also red, and then the blue are the arteries, and then in the notes it's also in blue, and then you can see how they are connected. Another way to remember how many arteries and how many veins, I always forget it, is you can draw a smiley face. Okay, the two A's are the eyes, so there are two arteries, and then the mouth is a V, which is the vein, so that makes a nice smiley face. Okay, so that can help you remember. This is a nice summary of also, if you've noticed, if you really look carefully, if you have your exam guideline next to you, you will see that we actually covered all of those bullets, and we spoke about the structure and function of all of the things that you're supposed to know. Let's go through it quickly so I can show you. We've got the chorion and the chorionic villi. This is also nicely color coded. The orange ones are the ones that go together, the chorion and with its chorionic villi. The amnion with its amniotic fluid, and then we've got the placenta over there, and then we've got the umbilical cord. And this is showing you, remember they said you need to know the picture of the fetus inside the uterus. So this is a lovely picture to show you, you need to know this. You need to know how to label it. Okay, what I want to bring to your attention as well over here is the other labels. Okay, remember they can ask you to label anything here. So you must understand that we have we have a uterine wall. Okay, so this is the muscle layer over here, the muscular part, that is the wall. Then we've got the lining, which is the endometrium, that's the darkened area over here. And then we've got the now we're looking at the placenta. We can see the placenta over there. You can see the chorionic villi that's become more vascular and larger. Then we've got the placenta they're showing you. They're showing us the outer layer is the chorion, the inner layer the amnion, and the amniotic fluid. Okay, and then we also have the umbilical cord. Okay, so you've got everything here summarized into one picture. Okay, then we've got gestation. Okay, before we go there, let's just recap quickly, okay, on what we did. So what have we discussed? Okay, we've discussed now that we had, once the ovulation has occurred, the ovum is in the fallopian tube, right? And it's waiting for fertilization to occur. The sperm cells are going to swim up past the cervix into the uterus, going into the fallopian tube, and then we're going to have, hopefully if it's successful, fertilization will occur. Okay, and this is when the head of the sperm cell enters the ovum, the tail falls away, and then the nucleus of the sperm cell fuses with the nucleus of the ovum. And now we are going to have a diploid zygote. That diploid zygote is going to still be in the fallopian tube. It's going to divide by a cell division called mitosis, and it's going to divide into a 2-cell structure, 4-cell structure, 8-cell, and then it's going to become a solid mass or solid ball of cells, which is called the modula, and then it's going to develop into a hollow ball of cells, which is called the blastocyst, and then it's going to travel down the uterus and it's going to implant into the endometrium. How does it do this? It's going to develop an outer layer, which is called the chorion. The chorion is going to develop chorionic villi. Those villi are going to extend or penetrate the endometrium, and it's going to allow the embryo, the blastocyst, to lodge in there. And then those chorionic villi are going to become more glandular, not glandular, it's going to become more vascular and larger. And then we've got the other structures we spoke about, the amnion, the umbilical cord, and then very important, the development of the placenta, which is going to be the endometrium with the chorionic villi. That is called the placenta. Okay, and in the endometrium we've got these large spaces that's going to be filled with blood, and that's going to bath the fetal part, so that the blood actually never mixes, right? But there's going to be diffusion between the maternal blood and the fetal blood, right? And then, as we spoke about, the fetus is going to need oxygen and nutrients, and then it's going to give off the carbon dioxide and the waste products, which needs to be taken up by the mother. And all of this is done via the umbilical cord that has one umbilical vein and two umbilical arteries. Okay, so that is just a quick summary, but lastly, just to remind us, the other definition you need to know is the difference between gestation and pregnancy. Learners always get this incorrect. Please make sure you know the difference between gestation and pregnancy. Gestation is the period of development of the fetus in the uterus, which takes place from conception to birth. But pregnancy refers to all the changes, the hormonal, the physical, the emotional, that take place in the body of a female because of the developing fetus. Okay, so that is the difference. Make sure you know it, because they like to ask this question, and learners always say pregnancy instead of gestation. Okay, and there's a definite difference, so make sure you know it. Okay, then that is all from me. I'm going to hand over to my colleague, Nonhlanhla Mshali, that's going to take you through all the possible questions that we can get in this topic. So please don't go away, and make sure that you can locate your worksheet that we have developed that goes with this PowerPoint presentation, so that you can also do the activities yourself while we are doing the lesson. Thank you very much.
You have been given power to decide whether you want good results in your life, in your schoolwork, or you want bad results. As Tony Robbins said, it is in the moment of your decision that your destiny is shaped. And your destiny is shaped right now with the decisions that you are currently making in your schoolwork. So decide whether you want to be an A student and start working on it, because there is power inside of you. The infinite power is inside of you, and the time you decide to tap in that power, you will become who you want to become in your schoolwork. Look at where you have been and ask yourself, am I happy with the results? Look at where you are right now and ask yourself, am I happy with what's going on in my life? What differentiates successful people from unsuccessful people, A students from average students, is the things that we do on an everyday basis. If you want to change your results, you need to take that one thing and repeat it over a period of time until it breeds results. And that is the art of consistency, because consistency breeds success.
Excellence. Excellence is a habit. Excellence is a quality you have, a quality I have. It's a quality we have when we honor what is true to us. Yes, we are gifted differently. uNkulunkulu akasinganga ngokufana. Excellence is striving for your best. Whenever you have a particular project, test, homework, uzibuze, ask yourself, is this my best? Can I put in more effort? Excellence is striving to be in the top 1% of the area in which you are gifted. And so I challenge you, in every single thing that you do, in every single area that you are gifted in, whether it's sports, it's academic, ucula, whatever that gift is, strive for excellence. Attitude of gratitude. What does that mean? An attitude of gratitude is the quality of being thankful. It's the quality of giving thanks. Ubonga, ulebuha. It's to be grateful. You and I have so much to be grateful for. You and I have parents who make sacrifices day and night for us to be at school. You and I have people who believe in us when we don't believe in ourselves, who love us when we don't love ourselves, who invest in us day and time again. Whether it's a teacher, it's a principal, it's a driver, it's a guardian, we have so much to be grateful for. And so I challenge you, young person, be thankful, ubonge, leboha, be grateful each and every single day of your life. Strive to display an attitude of gratitude.
Good afternoon, grade 12 learners. Welcome to the second session of today's lesson. From now onwards, I'll be taking you through the questions. We all went through the first part of the lesson, which was the content on the last part of the topic that you are currently busy with, which is human reproduction. And all the sections that Vedika covered, she went through them step by step. She even repeated how the process of fertilization, implantation, and pregnancy takes place. So now we're going to see if we can apply that knowledge that we've just received onto the questions. Remember, all the questions that I'm going to be doing with you are available in the worksheet that was sent to all the teachers. So make sure that you do get that copy. And then later on, when you are on your own, you go through the questions and you practice and try and answer as many questions as you can. Remember, if you still don't understand, your teachers are there. We are also here. We did give you the WhatsApp number when we started to say, do contact us if there are things that you still want us to discuss or if there are sections that you do not understand. So we're going to look at the first activity, which is activity number 1. So here we are looking at the diagram that shows the events that takes place in the female body. This is a very familiar diagram. Those of you who are used to practicing as many questions as possible when they study, you can already tell what is going on there because these are the diagram. This is the diagram that you've seen before so many times. So we always say to you, if you get questions like this in an exam, before you even look at the question, you study the diagram. So you study the diagram, you try and identify all the things that are happening there. If there's labels, you write down the labels so that by the time you get to the questions, you already have answers. It makes it so easy. So make this a habit. Each time you get a question with a diagram, when the question says study the diagram, that is an instruction. You must study the diagram before you answer the questions. So let's look at the first one. So we are looking at the diagram that shows the events that takes place in a female body, and we've got our labels there, A, B, C, D, E, F up until G. So if we are looking at A, if we're all looking at A together, we can see that those are the sperm cells, and what they are attached to, it is the ovum, right? So we would assume that what is happening there is fertilization. And then B, it was formed. This is what is formed after fertilization, and this is what is called a zygote. So this is our B. And then the process that it labeled at C, that results in that 4-cell structure there, this is what we call mitosis. Remember the difference between meiosis and mitosis. Mitosis results in the increase in number of cells, and it results in growth, whereas meiosis, it reduces the number of the chromosomes. And then with mitosis, these 4 ball cells form another cell that has many cells now in it, a ball of cells, which we call the morula, and then that is D. Then this cell continues to divide with mitosis, and it gives rise to a hollow ball of cells now, which is called the what? The blastula or the blastocyst. Remember, those are the differences that Vedika said. We have the blastula, we have the blastocyst, not the blastocyte. So do remember that. And then at E, we can say that is after implantation has taken place, and we are looking at the developing fetus in the uterus. So let's see if you can answer the questions now that we've analyzed everything that is taking place in the diagram. So the first question says, identify structure B. So if we look at structure B, we said when we were analyzing the diagram, we said structure B is the what? Is the zygote, because this is after fertilization has taken place. And then the next question says, identify structure D. We said structure D is that the solid ball of cells, which is called the what? A morula. And then Another one is structure E, and then we said structure E is a hollow ball of cells that is called a what? A blastula, or a blastocyst. So let's see if our answers are correct. So B is the zygote, D is the morula, the solid ball of cells, and E is the blastocyst or the blastula, which is a hollow ball of cells. Then the next question says, name the process taking place at A. So if we look at A, we are looking at an ovum, and it is surrounded by what? By a number of what? Of sperm cells. And then when Vedika spoke about fertilization, you said only once the head of one sperm cell has entered, has broken down this wall, and then this ovum then develops a protective layer which will prevent the other sperm cells from what? From getting in. And remember, it is only the head, because the nucleus is the what that is important here, needed to fuse with the ovum and with the nucleus of the ovum to form a what? A zygote. So the process that is taking place at A is what we call fertilization. And let's see the answer. Yes, the answer is fertilization. Then the next question says the inner wall of the uterus. So we're looking at the inner wall of the uterus where structure E implants. Because remember, once we have this structure, remember this whole process, this part here where fertilization takes place and this development through mitosis, it takes place in the fallopian tube. So it is moving in the fallopian tubes up until it is at this stage where it is ready for implantation, and where it implants itself, it's in the walls of the uterus, and that layer is called the what? The endometrium. So the inner wall of the uterus where the structure E implants, it is what we call the endometrium. Then 1.3 state the type of cell division. Now we're looking at the type of cell division that takes place at C. So we know, we are given a clue here. We are asked the type of cell division. The question did not just say name what is happening at C. You would have been maybe flustered a little bit as to what is happening at C. But here there is a clue. So this is one of those easy questions where you must take all the marks if they are presented to you. So there's only two types of cell division, and here we are looking at this one cell structure, and here we're looking at 4 cells now. So it is obvious this is the type of cell division which you call what? Mitosis. Increases the number of the cells. Then the last, 1.4 says how many chromosomes are normally found at H. Remember, we said H is the ovum, or this structure, this whole structure is the ovum, and the nucleus of the ovum has how many chromosomes? It has 23, because remember, it is formed by the process called meiosis, and meiosis, it is the reduction division. It reduces the number of chromosomes, or it halves the number of the chromosomes. So here it is 23. And then the last question for this part, it says identify the extra embryonic membrane at G. Vedika said a lot about the extra embryonic membrane. So the extra embryonic membrane that is found at G is what we call the what? The chorion. So do know the differences between these words, because later when we do another topic, you will find that there's a word that is almost the same as this one. So don't confuse the two. So here in the human reproduction, we speak of the chorion, we speak of the blastocyst or the blastula. So don't confuse it with the other terms that you will come across in future. So that was part 1 of the question. So I think this was easy stuff. We can all go through this. These are the types of questions that you will get, but it's not always easy like this where you get straightforward questions. Remember, life sciences is also more about application now. So you should be able to answer questions that are at a higher level than what we have here in front of us. So we said 23, and then the last answer is the what? The chorion. So those are all the answers. So I hope you've taken them down so that you can use them when you answer your worksheet. Then we have the second question, activity number 2. Here we are looking at the diagram that shows a developing fetus in the female reproductive system. So a A learner that knows his or her stories would have her highlighter or his highlighter underline all the keywords now. So we are looking at the developing fetus, and where is the developing fetus? It is in the female reproductive system. Again, we look at the diagram, we study the diagram before we answer the questions. So we are looking at the developing fetus. We can all see it over there. We know that this is the uterus. And then if we try and identify all the labels, we are looking at A, which is the extra embryonic membrane that is called the chorion. We are looking at B, which is the fluid that surrounds the growing fetus. That is the amniotic fluid, and it is in the amniotic sac. And we're looking at C, which connects the fetus to the mother, and this is what is called the umbilical cord. And we're looking at E, which is called the placenta. And then the last part there at the bottom, this is what we called the cervix. Remember your labels from the female reproductive system. So we've labeled everything. So we know now we are ready for the questions because we have identified all the parts that we labeled there. So the first question says, identify membrane A. So membrane A, it is that outside layer, the very, very outside layer, the outermost layer in the structure, and that is what is called the chorion. And then part D, if you remember your male female reproductive system, part D there at the bottom, it is what we call the cervix. And let's see if our answers are correct. So the first one is the chorion, part D is what we call the cervix. Then 2.2, give two functions of fluid B. Now, if you did not know what fluid B is, do you think you would be able to answer this question? So you see where I said the questions get the order, or they become more complex as you practice, as you look at the different questions. So here, now you must know first of all, you should know what fluid B is before you can give the two functions. So if you don't know what fluid B is, it means you won't even be able to give the functions and you have lost 2 marks. So here we're looking at fluid B. We all said that fluid B is what? The amniotic fluid. So what is the function of the amniotic fluid? We learned earlier that it acts as a shock absorber. It is for thermoregulation, prevents desiccation, also for what? For thermoregulation. And what was the other one? With the thermoregulation, remember, it maintains that constant body temperature, prevents the death of the fetus if there are changes in the temperature, and then prevents desiccation as well. Make sure that it's always moist, it does not become dehydrated, and also acts as a shock absorber, prevention from what? From mechanical injuries. So those were the labels, functions that we said. I might have left out some, but you do remember them, because I remember on the slide there were 5 that were written there. So it keeps the fetus hydrated, it keeps the fetus within the small temperature changes, that is thermoregulation, acts as a shock absorber, prevents mechanical injury, it allows free fetal movements, and those are the 4 functions that are there. But remember, the question said only 2. So those are some of the functions that you can write. Now, the next part, I'm still on the same diagram. Now the question says, explain how the development of the fetus would be affected if part C became completely blocked. So what is part C? Part C is the umbilical cord. Earlier, we were told that the umbilical cord has 1 vein and 2 arteries on it, and those are very important. The vein, what does it do? It brings in the oxygenated blood, it brings in the what? The nutrients, whereas the arteries, they take away the waste products and they take away the what? The carbon dioxide. So it carries the deoxygenated blood. So now, if this part were to become completely blocked, what would happen to the fetus, to the development of the fetus? So now this is what we need to explain. So it means that number 1, there would be the fetus will not be able to receive nutrients from the mother. It will not be able to receive oxygen from the mother. And 2, it means the carbon dioxide from the fetus and the waste products from the fetus will not be able to be taken from them back to where? To the mother. So this might cause poor development, which might also lead to what? To death. So you see here, we've mentioned the cause and the reason. So what would happen? So there's two things. So remember, with the explain, it is the cause and the effect. So it will cause the nutrients not to reach the fetus. It will cause oxygen not to reach the fetus. It will cause carbon dioxide not to be taken away. It will cause waste products not to be taken away. And what will be the effect of this? The effect will be slow in development, and which might even lead to what? To death. So those are two things. Those are your explain question. We're looking at the cause and we are looking at the effect. This is how you get your 3 marks. So you see how I did not just give one sentence. The question said explain. My answer was not just one question, one sentence. Also, I did not give the function. That is what another mistake that we do. When the question says if part so and so is blocked, we are very quick to just give the function and not answer the question. We are not giving the function, but yes, you must know the function to answer the question. That function and the effect if that function cannot be carried out. So let's look at the answer. Okay, here it says nitrogenous waste will not be excreted and will accumulate in the fetus, resulting in slow underdevelopment or even death. You see how the 3 marks came about? It's nitrogenous waste that will not be excreted because the arteries will be blocked. Then the nitrogenous waste will accumulate in the fetus. And what will this do? Remember, the question says how the development will be affected. It will result in slow or underdevelopment, which might eventually lead to what? To death of the fetus. Or oxygen and nutrients will not reach the fetus, leading to poor or no development or suffocation in that case. Then this might also lead to what? To the death of the fetus. The 3 marks. So please be careful when you answer questions. Read, understand before you answer. Also, we always say to you, do not leave questions unanswered. So you're going to attempt all the questions when you get the question paper. Okay, moving on. The next question says, name the 2 systems in the human body whose function is served by region E during gestation. Remember, gestation being the period of development. Know your differences between gestation and pregnancy. During the period of development of the fetus, that is gestation. So what systems? Now this one says name 2 systems in the human body whose function is served by region E. Region E, the whole of region E, we said this is the placenta. Remember, there's chorionic villi from the fetus that embed itself or implant themselves on the what? On the endometrium in the uterus, and then together the endometrium and the chorionic villi from the fetus, and that whole structure is what we call the placenta. So what are the 2 systems again that are represented by the placenta? There's more than 2, but the question just asks you for 2. So remember, we said the function of the placenta, it acts as a point where gaseous exchange takes place, where exchange of what? Of nutrition takes place. It provides immunity to the developing fetus. And we also said it produces what? Hormones that will maintain pregnancy. So what systems have I mentioned? I've mentioned gaseous exchange. So we're looking at the gaseous exchange system, the breathing system. We talked about nutrition. So we are looking at the digestive system. We spoke about hormones. Then we are looking at the endocrine system. We spoke about immunity. Then we are looking at the what? At the immunity system or the immune system. One would ask, why are we talking about the immune system? Remember, with the developing fetus, this part E acts as what? It acts as protection. And then during the last months of pregnancy or of gestation, the last 3 months, the last trimester, that's what they call it, that is when there's antibodies that will come from the mother and they will be transferred to the growing fetus. That is what you studied in grade 11, what we call passive immunity. I don't know if you still remember that, but then you can link this with your grade 11 word, passive immunity, when the antibodies are transferred from the mother to the growing fetus. That is why we speak of the immune system. Then the last question says, describe the development of a fertilized ovum until implantation occurs. So here it's this, you describe. You are not explaining, we are not looking for the cause, we are not looking for the effect, but we just need to know what happens after an ovum has been fertilized, the development. So we want the process, the steps, the sequence of events as they plan out during the development of a fertilized ovum. So what are we looking at here? We're looking at the zygote. Remember, Vedika said at the beginning, after fertilization, the diplozygote is formed, where the nucleus of the sperm cell fuses with the nucleus of the ovum, and they come together and they form the what? A diploid zygote. And then we said, in that first question that we did, we said then that zygote divides by mitosis, and then it forms what? A ball of cells, which is called a morula. And then we said that solid ball of cells continues to divide by mitosis, and then it gives rise to a hollow ball of cells, which is called what? A blastocyst. So the question said until implantation occurs. So that is why we stop at what? At blastocyst, because this is the structure that implants itself on the uterus in the female reproductive system. So that was then activity 2. I hope you are able to connect the questions with the first part of the lesson, which was the content. Now, activity 3. Again, I told you that this diagram looks familiar. The first activity that we did, it was this diagram, but the first one was in the circular form. So here we're just looking at the straight line diagram, but it's exactly the same thing that we saw earlier. And another thing that is different here is that the parts are mixed up. Whereas with the first one, it was following the correct sequence. That is why I said to you, there was the simple question, but now we're doing more questions that are becoming more complex. So you should be able to put these in order so that you can answer the questions. So it says, it shows the events again that takes place in the female body. So we have number 1, which is the ovum. At least we are told what this is. Number 2, we are not told what this is, but we would assume that because we are looking at the sperm cell over there, so obviously this would be what? An ovum. So what is taking place here, and no one needs to tell us, we can all see that here what is taking place is fertilization. And then if we're looking at 3 looks like this cell over here. So 3 is still a what? We can still say it is a developing what? Ovum, because it looks the same as this one. And then, or this is after now fertilization has taken place. And then 4, we are looking at the solid ball of cells. Remember the solid ball of cells? So 4, we are looking at the morula. So 3, we can say 3 is after fertilization is taking place and say zygote. And then the last one is labeled, at least we know this one is called the what? A fetus. So identify the reproductive structure at 3. So you see what I said now? Then this is where we see that, okay, whatever we did there, the labels were wrong. Initially, we said this, it looks like a what? The ovum before it is what? Before it is fertilized. But now the question says, identify the reproductive structure. So you see the examiner's throwing you off there. So is it a zygote or is it a what? An ovum? But because the ovum is labeled as number 1, there's no way that this can be a what? The ovum again. So we'll keep to our answer and say what is 3 is the zygote. And then stage 4 is obvious. It's the solid ball of cells, and this is what we call the morula. So let's go back again. We are saying what is represented in stage number 3 is the zygote, because we are saying this has already been labeled there. So this is the zygote, and then this is the morula. So let's see, answer number A is zygote and answer number B is the morula. So we are correct. Then 3.2, name the process that is taking place at stage 2. The process. The diagram is clear. You cannot go wrong there. The process is fertilization. And the last question says, where in the body does the process at stage 2 occur? So where in the body does this process takes place? We said in the first question, it takes place in the fallopian tubes. So that is your answer there, and that is your 1 mark. Moving on the very same diagram, name the process responsible for the formation of the reproductive cell shown at stage 1. So stage 1, this is the ovum. What is the process that is responsible for the formation of this? It is meiosis. Still remember that? And you see that these questions are more or less the same. And yes, it's meiosis. Now 2, describe 2 ways in which the fetus is protected. So the first one, we said it's meiosis. Okay, now let's go back. We said our answer is meiosis. We are not wrong, but we can see here that we needed to give a more specific answer. Because why were we giving a more specific answer? Because this is the key word here. We are told that this is an ovum, and the process that gives rise to an ovum is oogenesis. It is meiosis, but what type of meiosis is it? It is oogenesis, because the one that results in the formation of the sperm cells, we call it the spermatogenesis. So describe 2 ways in which the fetus is protected. We spoke of the amniotic fluid. We also spoke of the placenta. So the amniotic fluid, remember all the functions in that slide where we said it acts as a shock absorber, it protects the fetus from mechanical injury, it controls the temperature changes, or it helps when there's temperature changes, it prevents against dehydration. And then we said there's a placenta which serves as a barrier protecting the fetus from what? From any diseases that might come from where? From the mother. And then the other question is still on the development of the fetus in the female body. Here we're looking at there's only one label here. We're looking at the fetus and we're looking at the structure. We can all now identify the structure. This is the placenta, and this, what connects the placenta to the fetus, this is the umbilical cord. So we do know that now. We are clear. And Vedika spoke about the chorionic villi. We can see the chorionic villi that are embedded onto the endometrium, and then this is why we say this whole thing is a placenta. 4.2, tabulate 2 differences between substances transported in the veins and arteries of structure A. Remember, umbilical cord, veins, and arteries. Tabulate the differences between the substances that are transported. Veins, umbilical vein transports oxygen. Umbilical vein transports nutrients. Umbilical artery transports carbon dioxide. Umbilical artery transports what? Waste products. So, or you can say carries deoxygenated blood, the other one carries oxygenated blood. So let's look at the differences. Okay, the first answer there is umbilical cord, and the answers are here. Carries deoxygenated blood, carries oxygenated, deoxygenated, carries nutrients, and this one carries waste products. Remember, with the table, you get 1 mark for drawing the table, and then you get the other 4 marks. So it is important. You draw your table, you put the headings. Do not forget to put the headings, because if these are not here, then you are leaving it to your teacher to assume that, okay, the first column of your table is umbilical vein and the second column of your table is artery, and you will be marked wrong. So you must be specific. Put the headings at the top so that you get all your 5 marks. Those are the rules when you answer any question that says tabulate. You draw your table, neatly so that you can see that it is a table, and then you put in your headings for the columns. Then you write the correct differences, and your differences must match. You see here we spoke about it carries oxygenated blood. This side carries deoxygenated blood. The answer here was not carried waste product, because now we are talking about two different things. So if your first side, your left-hand side speaks about oxygenated or gases, then it means on the other side you must also talk about gases. Here we are talking about nutrients, then on the other side talk about waste products. So be mindful of that. And then the two functions of the placenta, again, this was mentioned: gaseous exchange, nutrition, immunity. We spoke about the immune system. We spoke about the it producing the hormones that are required to do what? To maintain pregnancy. So it's nutrition from the embryo, excretion, gaseous exchange. It also acts as a microfilter, preventing anything that is harmful for not reaching the what? The fetus. So you'll see that some of the questions now are moving fast because we've already covered them. So there's no need for us to go through them again in detail. We've already done functions of the placenta before. Again, I think this is the last question now. Identify, okay, now I'm doing it all wrong. I said to you earlier, we study the diagram, we identify all the parts in the diagram before we answer the questions, no matter how tired you are. You see, now I was tired and I was jumping to the questions, and that's not what I said to you. So let's go back and do the right thing. So diagram below represents the relationship between the blood system. So we're looking at the relationship between the blood system of the fetus and that of the mother, and the arrows indicate the direction of the blood flow in the vessel. Sorry that this is not clear, but this is vessels there. So we are looking at the relationship between the blood system of the fetus. Here is the fetus represented by this circle, and here is the placenta here, and that of the mother. And then there's arrows that represents the flow of blood. So there's blood vessels that are going into the uterus and coming into the fetus, and the blood vessels from the fetus back to the placenta to the mother. So this is all that we are looking at. And we said P, it's the chorionic villi. And we said this is blood vessel A because it goes to the fetus. So blood vessel A is the umbilical vein. And then that is the one that is moving away from the fetus to the placenta. This is the umbilical artery. So this whole structure here, we'll say it represents the umbilical vein. And then this is the placenta. And then these are the blood vessels of the mother now. Blood vessel D and blood vessel C are the blood vessels of the mother. So now we must identify structure P. We can see its finger-like projections and it embedded onto the endometrium. So structure P is the chorionic villi. Extra embryonic membrane, remember we said the extra embryonic membrane that gives rise to structure P, we said that is the chorion. Then again, the functions of the placenta, acting as the microfilter, providing immunity, gaseous exchange, nutrition, and what else? Yeah, we'll just look into them now. So let's look at the answers. Chorionic villi, number 1, so we are correct. Chorion, extra embryonic membrane, and then the functions of the placenta: acts as a microfilter, prevents harmful substances from reaching the fetus, produces antibodies, remember the passive immunity that I discussed earlier, secretes hormones, which is progesterone that maintains the endometrium, also immunity that is transferred. So this is the same, it's the same as that one, so it's just repeated twice. Then this is the part 1 of activity number 5. Then part 2 of activity number 5 is that we are told that blood vessel A is the vein. We've already said, because the arrow is going here, this is the vein. So tabulate the differences now between the composition of blood found in A and blood found in B. We've already answered a question like this. Let us just go straight to the answers. So here, the only difference here is that you are not told what blood vessel A is and blood vessel B is, but the arrows are there to guide you This one goes into the fetus, so it is the vein. This one is going away from the fetus, therefore it is the artery. So this is what you do. You write your blood vessel A, blood vessel B. You see what this answer did? This and the examiner here did not write umbilical vein and umbilical cord, because this instruction says between blood vessel A and blood vessel B. So your headings are blood vessel A and blood vessel B. That is what is expected. And then the difference in the composition now, that is another difference. That other one asked us what is carried by the umbilical vein and what is carried by the umbilical artery. Here we are looking at the composition of the blood. So here we are looking for these terms here. There is high concentration of nutrients, there is low concentration of nutrients. There is low concentration of waste products in the vein, there's high concentration of waste product in the artery. Low concentration of carbon dioxide in the vein, high concentration of carbon dioxide in the artery. High concentration of oxygen in the vein and low concentration of oxygen in the what? In the artery. So those are the differences. So when you answer these questions, be careful of the statement that is provided. Composition, and then that side it was what is carried by this and what is carried by that. So it was safe for us to say it carries oxygen, it carries carbon dioxide, it carries nutrients, it carries waste products. But here, the difference in the composition now. So we are looking for that high concentration and low concentration of substances in the blood. 5.4. Explain one consequence for the fetus if blood vessel B become blocked, preventing blood flow. So blood vessel B is the umbilical artery carrying carbon dioxide, carrying waste products away from the fetus back to the mother. So if it becomes blocked, it means, remember the cause and the effect. If it becomes blocked, what will it cause? It will cause the accumulation of the waste product and carbon dioxide. There will be a buildup of carbon dioxide and waste products that cannot be transported in the fetus. And what will be the effect of that? Effect is that it might lead to death, it might lead to slow development or abnormal development of what? Of the fetus. So let's look at the answer. Yes, waste products, nitrogenous waste, will accumulate in the fetus's body, causing death or harm to what? To the fetus. So we are looking at the cause and we are looking at the effect. Then here, this question, I want us to look at this. There's a part that is missing in this question because it says, describe how this could lead to death of the fetus. So there's a part that was missing, which is just picked up now. So the instruction for 5.5 was, if the blood, I hope you are all listening, if the blood of the mother and the blood of the fetus come into contact with each other, it could lead to death of the fetus. Regika did speak about the two, that they do not mix, and the reason why they don't mix, because they might lead to death of the fetus. So now the question says, describe how could this cause the death? Describe how mixing of the mother's blood with the fetus's blood will lead to death. So remember what we said earlier. When we look into this, we said they do not mix because the placenta is there to make sure that the harmful substances from the mother do not get into what? Into the child or into the fetus. For example, let's say if the mother is sick with flu, for example, so the bacteria or the virus that the mother is carried that time is prevented from reaching the blood's child, because if that would happen, if it reaches the fetus blood, then it may lead to death. So how this could lead to death? Harmful substances may be passed from the mother's blood to the fetus's blood. So that is one thing. There might be harmful substances that might be passed. 2, their blood types might not be compatible. So if the blood types are not compatible, that would also lead to death. Remember the compatibility story? You will learn more about this when we do genetics in the next chapter. So that is why again we say they cannot mix and it might lead to death because of these reasons: harmful substances and the compatibility of the what? Of the blood types. There was one more question that is in your worksheet. Here we were just looking more at the what? At the skills. Remember we say that skills are also important. So it is important that with all the questions that you practice, you also look into the questions where you draw graphs, you draw diagrams, you do a little bit of calculations, you look at the scientific investigation skills, because that is where also 20% of your question paper comes from. So we have a question here. This is our last question, I promise. I did say it's the last one, activity 5, but this is the last one now, I promise. Activity 6. It's a story that we must all read together. We're looking at an investigation that was conducted to compare the reflex action in new babies with premature babies. So we're comparing reflex actions. This is what is being compared. Reflex actions. Between what? Between newborn babies and premature babies. So the premature babies are those that are early born and not carried for the full term. So they are not carried until the 40 weeks of gestation. They might be born maybe on week 30, week 28, week 25. Those are what we call the premature babies. So a newborn baby changes of survival is evaluated. So they evaluated the chances of survival by a doctor, and the score that was found there was recorded on the AGA scale. So what the doctors looked for, they looked for the heart rate, they monitored the heart rate, they monitored breathing, they checked the muscle tone, the color, reflexes. All of these were used as the criteria for this test. Then the following reflexes were tested: the palm grasp, sucking, swallowing, moving of the head in the direction of touch on cheek, and the walk reflex, walking movements made by the legs of the baby. Then the data that was collected from the 1,500 babies over 2 years is shown in the table below. So we are looking at the table, the gestation period, the period of development, which is normally from week 1 up to where? Up to week 40. And then the number of babies that were tested, and then the results. This is the reflex time that we are looking at, specifically this one from the palm grasp, and then the score that was recorded on the AGA, on the AGA scale. So if we look into this, now we're looking at the reflex time, the amount of time it took. So we can see that premature babies between 24 and 27 weeks, their time was longer than when you compare the babies or the fetus that was carried to full term up until week 40. The time became less, so their response time was less, and that was 1.9. Remember, when we talk about the reflex action, you learn about that in the nervous system, how you respond, that involuntary response to any change that takes place in an environment. It is a quick involuntary response. You will learn more about it. So full-born babies, they react faster than premature babies. You can tell by the time. There's 4.7 here, there's 1.9 there. So let's look at the questions. Identify the independent variable. So here we just want to highlight independent variables and dependent variables. Remind you, because you've done this in grade 10, you've done it in grade 11, in grade 12 it's just a repetition. Remember, independent variable, it's what you can, okay, we have the dependent and we have the independent variable. Dependent variable depends on the independent. So what you cannot change, the gestation period. We have the gestation period of what? Of 24 weeks here that is mentioned up until week what? Week 40. So here, this is the independent what? Variable. And the dependent variable in this experiment will be the average palm grasp time of what? Of the reflex time. So this depends. This depends on the week. Like we said, your premature fetus or babies, they take longer to respond than the baby that has been carried on on full term. So the answer for this question is the gestation period. Then the next question says, from the data and the information given, draw a conclusion about the average palm grasp reflex time. So here we want to highlight one thing. When you speak of a conclusion, remember you must include both your variables. Your conclusion must show the variables. So we did say earlier, by just looking at the table, we said we can say the longer the gestation period, the shorter the reflex time. Or the shorter the gestation period, the longer the reflex time. So both variables are there, gestation period being the independent and the reflex time being the dependent variable. So let's look at the answer. The longer the gestation period, the faster the palm grasp reflex, or the shorter the reflex time. Or the shorter the gestation period, the slower the palm grasp reflex, and the longer the reflex time. Now the last question, easy question, draw a histogram to represent the results. What are we looking at? We are looking at the gestation period and the what, and the between the what periods, between the 27 and what and 37 weeks. So that is what we also must be careful at. We are given about 1, 2, 3, 4, 5 periods here, but we are only asked to draw a graph of the periods between 28 and 37 weeks. So it means your graph must only focus on these three. We exclude that and that. That is how you read the question. So some of you don't read. You just go straight to drawing without even checking the question, and you draw all the bars that are not required and you lose marks. So here we are only focusing on this period. So we are drawing and we're looking at the score. This is against the what? The average AGA score again. So what we are supposed to draw is this versus the score. So there are many things that are given here, but you are only asked to show two things. So this and that. This was quite a complex graph because you needed to read carefully for you to answer. So what we want to highlight here is that, remember the differences between the histogram and the bar chart. I'm just going to skip on to the next slide. Differences between the bar and the histogram, the bar graph. The histogram, the bars and the histogram, they touch, you see? There's no space in between. So that is what we're looking at. That 1 mark for type of the graph is when there are no spaces between the bars. And then you get your 1 mark. And then the labeling of, okay, the correct caption. So the correct caption is the average AGA score of babies with gestation periods of 28 to 37 weeks. Again, we're looking at two variables, the score and the period. So you get your 1 mark. Then the correct label for the unit of the X and the Y axis. The gestation period is in weeks, so we have the label and the unit. We have the label, the average, and what units it's out of 10, and again you get your 1 mark. Remember, both of them must be correct for you to get a mark. And then the scale of the X and the Y axis, we look at the bar sizes here, they are all equal. So it shows that the scale that was chosen are also correct. And then here we started from 0 and it increments by 1 up until we get to 8. There are no mix up of numbers here. We did not say 0, 5, 7, 8, 9, 10. All the numbers incremented by what? They increase by 1. So that is the correct scale that was chosen, and both of them must be correct for you to get a what? A mark. Then lastly will be the plotting of the points. So if all the points are plotted correctly, you get your 2 marks. But if you missed one, then you get 1 mark. So this task was just to remind you that, remember, when you do your practice from the past exam question papers, also look into the questions that are assessing the skills. So we have, I told you that this was the last, I promised that it was the last, it is the last activity. We have come to the end of today's lesson, which is the end of the human reproductive system. and the end of the Thursday lesson for this term. Yay! It means the schools are closing next week. So we will see you in term 2 with a new topic, which is genetics. And we are wishing you all the best. Please don't forget to study during the holidays. SIP is coming. Attend those SIP classes, attend the camps, practice, and ask your teachers if you still don't understand. We've come to the end. Thank you very much for your attendance. Bye.